I'd like to take this time to introduce our keynote speaker, Dr. Stephen Thomas. I'm honored to introduce our keynote speaker. He's the Chief of Infectious Disease Division, Upstate. He was recently appointed lead principal investigator for the Pfizer BioNTech global phase of COVID-19 vaccine trial. He's played a key leadership in the roles in the U.S. government response to the Ebola outbreak, as well as the MERS and Zika epidemics, and was instrumental in the development and advancement of vaccines for each. During the pandemic, Dr. Thomas has led SUNY Upstate's Incident Command, and his team has been at the forefront of experimental treatment and trials for COVID-19. If you have any questions for Dr. Thomas, please leave them in the comment chat box. Good evening, Dr. Thomas, and thank you for joining us, fresh off your appearance on CNN.
Thank you very much. No, it's really great to be here. This is my first society meeting, and I'm a proud new member. So that's probably one of the best things I did this week so far. So I know I'm conscious of the time, so I'm going to share my screen and get right into it. And if Justin, can you give me a thumbs up? Can you see this?
Yes, we can.
Okay, fantastic. Okay, so I wanted to talk about COVID vaccines today, and just some disclosures which were already mentioned. Okay, so what I wanted to do is to just kind of level set everybody's understanding about how the vaccine development process historically has worked, what the targets are for COVID vaccines. I'd like to give a little bit of the kind of the landscape that's out there, the different technologies that are being applied, and then I'm going to sort of do a deep dive into the Pfizer data. Obviously, today was a big meeting with the FDA. That was about a 9-hour meeting that we had earlier today, and ended up in I'm not sure if people are aware, but it ended up with the advisory committee advising 17-4 with one abstention to issue the emergency use authorization. So that was good news. And then we can do a little question and answer. So this slide's about 19 years old, but I still think it's a great slide because it gives so much information. It's almost like a dashboard. So when you identify that you have a problem, a public health problem, let's say it's COVID in this case, right? You got a bunch of groups that are going to be sort of in this discovery mode. And so you have all these potential candidates that are in the discovery mode. They're trying to figure out sort of, okay, what's the pathophysiology of the disease? What's the human immune response look like? Is it a protective response? How could we potentially, mimic this through vaccination, let's say? And typically, as you see here, and you can multiply it by whatever factor you need to get to 2020. But that's usually like a 3 to 7 year process that all this discovery is going on. And it usually runs about, $150 to $200 million. And about 1 to 3% of all these candidates actually make it into the next part of the process, which is preclinical development. Preclinical development is animal studies. So these could be small animals like mice or rats or rabbits, for example, and then goes up to larger animal studies, so non-human primates. And this preclinical development, this lasts about 2 years or so. It's a little bit less expensive, maybe $70 to $100 million. And of those candidates that make it into this phase, maybe 15 to 20% of them will then actually make it into the human testing phase. But there's a really big milestone here, meaning let's say that it's a vaccine and it's a laboratory-derived vaccine. You have to go through a biomanufacturing process. And this is important because, one, there are not enough biomanufacturing facilities on the planet, and two, so there are long queues, and two, it's incredibly expensive. And so a huge chunk of the $10 billion in Operation Warp Speed are to pay for biomanufacturing. So once the candidate actually kind of traverses this first sort of valley of death, if you will, and they get into human trials, there's 3 different phases typically. There's a phase 1, which is primarily focused on safety. It's a small number of people. Phase 2, also safety is the focus, but you really start looking in depth into immune responses. And this might be where you start looking at dose and schedule and how you deliver and what the whether it's 0-3 weeks or 0-4 weeks or things of that nature. And then once you can demonstrate in a larger number of people, so this could be hundreds of people, that the proof of concept that this is not only safe, but it generates the type of immune response that we think will be protective, then you can go into these phase 2B or these phase 3, big studies, tens of thousands of people. This whole process, as you can see here, this could take, 6, 7, 8, 10 years and cost hundreds of millions of dollars. And as you can see, more and more candidates are sort of falling out and they're failing. This is, again, another big milestone here because typically when you manufacture vaccines back here, you're manufacturing lots of tens of thousands of doses maybe to support your human trials. Here, you start manufacturing in the millions of doses, and the process is different and the regulatory requirements are different. But if you make it through here, then this is when you engage the FDA. So the whole thing can take 10 to 20 years. It's well over $1 billion US, and the success rate is about 1%. So keep that as your context for what has just happened with this Pfizer product, which I'm going to talk about tonight, which really went from here, preclinical development, to now here in 11 months. Okay, so SARS-CoV-2, we all know it's the virus that causes COVID, and this is what the virus looks like. If you were to cut the thing in half, then what you're seeing, of course, here is the RNA, the genetic material. You got this nice kind of lipid envelope that's around that genetic material, and then you have these glycoproteins which are sticking off of it, and these are the spike glycoproteins. And this is the business end. This is what we're going to be talking about through the most of this lecture. This is a blown up version of what spike proteins might look like. And again, this is that lipid bilayer. It's right here. Why this is so important is because it really is the primary mediator of this virus attaching to a target cell, like could be in your respiratory tree, for example, and is what creates the infectious process. So here's the virus, here's the spike protein. It's actually a trimer. And what it does is it attaches to this host cell. Again, let's say that this is a lung cell, and these lung cells have ACE2 receptors. So it's kind of like a key into a lock. And once the key goes into the lock, there are these conformational changes that ultimately result in the virus fusing and getting into the cell. And so that's what it looks like here. So the spike protein, it's a trimer. When it sees this receptor, it changes its conformation, it attaches, and then that cell is going to be infected. And once it's infected, what happens is it gets into the cell, it releases its genetic material, it goes through this process of replication of the genetic material, and the virus uses the cell's own machinery to make lots of baby viruses, and then those baby viruses go out and they go out to infect other cells, and this is where we get viral replication occurring. Now, one of the problems, if you remember, way back to, almost feels like almost forever, back to January, February, everyone was initially focused on the lung pathology that was being seen with COVID, right? And you remember the CDC's, here are the types of symptoms that you have to be concerned with COVID, and it was predominantly respiratory. The thing is, these ACE2 receptors are throughout the body. They're in the lungs, right? They're in the liver. So we see with COVID patients, we can see people with hepatitis. They're in the kidneys. We have a lot of people that end up getting acute kidney injuries. Some people end up having a complete renal failure, need to go on dialysis and things of that nature. You remember when the CDC was adding GI complaints, so people with nausea, vomiting, or diarrhea. The heart, so we would have patients that would get infected, get in the intensive care unit, make it through their respiratory problem, and then get up to the medical surgical unit, medical surgical ward, and they have some kind of fatal arrhythmia, because we know that the heart has ACE2 receptors. And we know we see people who lose the sense of taste, the sense of smell, so we think that that's ACE2 receptors in the brain. So anyway, you get the point. This is a really bad disease because the receptor for this virus is everywhere. So what would be a good goal then, right? This is that discovery kind of process. The goal would be, if we can develop antibodies against that spike protein, we could block infections and we could prevent disease. So that's the goal. So if we can do this, what are the desired outcomes of vaccination? And I know this group knows this, but one, we want to reduce infections. If possible, we'd love to have a vaccine that could reduce transmission. We, of course, want to reduce clinical illness in people because we really need to protect the healthcare system, right? That has always been one of our main concerns about COVID, is that the healthcare system, which already operates in this country at 90% capacity, would be overrun. And then we want to support our economy, education, and improve our quality of life. So the coronavirus vaccine tracker as of this morning, you can see here, so early studies, over 40 candidates, phase 2 studies, 17, more than a dozen vaccine candidates in these advanced phase 3 trials. And this is global, by the way. 6 vaccines that have been given limited approval, and then 1 vaccine that has been approved for full use. So what are we trying to recapitulate here with vaccination? Well, let's look again, when people get infected naturally and they develop immune response, do we believe that they are protected from subsequent infection? There are certainly case reports of people getting infected a second time, but there's also a number of reports that, that seem to demonstrate that people who get infected and develop a neutralizing antibody response are actually protected from reinfection. So again, virus enters the body, and when the virus enters the body, these virus killing cells, these antigen presenting cells, our immune system basically recognizes it as foreign, and this very complex orchestration of humoral and cellular immunity create antibodies and and T cell memory, CD4 and CD8 T cell memory, and we believe this protects people. So we want to try to mimic this natural immunity, one, because they think it protects people, 2, because we don't really believe that immunity at this point. We don't have the evidence that shows that immunity would increase your risk of a subsequent disease if infected a second time. Think of something like dengue, for example. And 3, because we think it's feasible. We think we can do this. So here are all these different vaccine approaches. This is the one landscape slide I was going to tell you about. So we have viral vectored vaccines. We have DNA-based vaccines, kind of traditional live attenuated virus vaccines, recombinant protein-based vaccines. And then here's the messenger RNA vaccine. So that's the 2 leading candidates right now, Pfizer and Moderna. These are both messenger RNA vaccines. Now, regardless of what type of approach you are taking, the outcome is the same, which is you want to create an effective and durable immune response, preferably both arms of your immune system, antibodies, so humoral, and your T cells, your CD4 and CD8 immune responses. So the way it works with RNA vaccines is you have the genetic code here for that spike protein, right? So you take this messenger RNA, they encapsulate it in this lipid nanoparticle. It gets you get the injection. It goes into the cell, so it goes into the cytoplasm. It doesn't go into the nucleus, and that messenger RNA code, it starts to replicate and it starts to produce these spike proteins. And these spike proteins go out into the body. The body recognizes them as foreign, and it starts to mount that immune response that I just showed you on the previous slide. These spike proteins, these are not live infectious virions. So these are not going to multiply. They're not going to cause an infection. They're not going to cause that person to be infectious to other people. But that's basically the mechanism of action of these vaccines. So I'm going to dig into the Pfizer vaccine because at least in the United States, well, it's been approved. It's received emergency use authorization in the UK, in Canada, and it's probably on its way to receiving an EUA in the United States as well. So we published this morning in the New England Journal. So this is a phase 3 trial. 44,000 people were screened, and ultimately about 43,000 people underwent randomization. Those people then either were randomized 1-to-1, either to receive the vaccine, which is this BNT, so BioNTech 162b2, or they were randomized to receive placebo. And then 37,000 had a median follow-up of 2 months, which was a requirement from the FDA prior to any sponsor being able to go to them and ask for an EUA. That 2-month mark comes from the fact that there's a lot of data that supports that if people are going to have some kind of immune-mediated adverse reaction to vaccines, it typically occurs within the first 6 weeks. So that's why they chose that 2-month mark. So you have 18,000 receiving the vaccine, about 18,000 receiving placebo, a number of people, about 300 in each group, for a host of reasons, and this is pretty typical. It's actually a pretty low percentage for a trial of this size. Another handful of people in both groups were discontinued the trial after dose 2, but this is basically how the trial was randomized. And this is the sort of the time and events table. So 2 doses, 21 days apart, active surveillance for COVID-19 illness for the entire follow-up period, which is going to go for 2 years. 7 days after each dose, we tracked very closely 6,000 people in this sort of reactogenicity. So we wanted to look very intently at 6,000 people across the different countries to see what kind of reactions they were having and describe them in detail. And then we follow people, all subjects, for serious adverse events, and of course we track deaths and any serious adverse events related to vaccination in all subjects throughout the trial. So here are the demographics. So males and females were split pretty evenly. The vast majority of volunteers were white, so 83%, had about 9% Black or African Americans, 4% Asian, and then you really get into very small numbers in some of these other groups. This is really, it's unfortunate, and it was something that was mentioned today at the FDA meeting, that the percentage in non-white groups is pretty low, and it's unfortunate because these groups, as we know, bear a much higher burden of illness and hospitalization and bad clinical outcomes, to include death. About 28% of people were Hispanic or Latinx. Most of the volunteers were enrolled in the United States. Argentina, Brazil, South Africa were also involved. And then you see the breakdown in age here. So 16 to 55-year-olds, about 60%, and greater than 55, about 40%. And then you can see here about 35% of the subjects in the reactogenicity index were obese, having a BMI greater than 30. So this is just looking at local events, so the injection site, and you're looking at pain, redness, swelling at the injection site. This is the percent of subjects who experienced it, and the different colors represent: green is mild, blue is moderate, the orange is severe, and red is grade 4, which is life-threatening. So what you see is, and this is the 16 to 55-year-olds, so what you see is that the rates of redness and swelling are low, and they are comparable to what we have with licensed vaccines. And then we have a significant, we have more people reporting pain in the arm with vaccine compared to a placebo. The vast majority, though, is mild to moderate. What that means is I have pain in my arm, but it's not impacting my life. That's mild. Moderate is I have pain in my arm that's limiting some of my daily activity, but I can still do what I need to do: go to work, take care of my family, go to school. Severe is when you start having a reaction that actually impacts that prevents you from doing those types of activities. And then the greater than 55-year-olds, they also have the same kind of pattern. But what you'll see here, and this is a consistent trend, the safety profile seems superior in people over 55 compared to those between 16 and 55. Now, this is looking at systemic complaints. So we're looking at fever, fatigue, headache, chills, vomiting, diarrhea, muscle pain, joint pain, and then any kind of use of any antipyretics. So again, dose 1, dose 2, 16 to 55-year-olds for both of these. And so what you see is that the most predominant adverse event that is being tracked is fatigue and headache. That's the most common. We also see chills. We also see some muscle pain. GI complaints much less, and then we see some fever. Again, though, it's mild to moderate. That is the predominant. That's the predominant grading that these volunteers gave for the symptoms that they were having. The other thing that I'll tell you is that these symptoms occur very quick. So most people, they're vaccinated on day 1. Most people start having their symptoms on day 2, and most people have resolution of their symptoms within 24 hours. So it comes on quick, it goes away quick. And this is for the greater than 55-year-olds, and it's the same thing, but again, it's flipping back and forth. You can see it's less. It's less than the younger age group. Okay, so then moving on here. So now we're looking at vaccine efficacy. So this is vaccine efficacy against COVID. When you start the counter, you start it 7 days after the second dose, okay? This first row up here, so we have the vaccine group here, we have the placebo group here, we have the number of cases in each group. So this is COVID that occurred in participants that did not have evidence of a past or current infection at the time they were immunized. So we did not enroll anyone who had a known history of COVID in the past. So we would ask the volunteer, have you ever been diagnosed with COVID? And if they said yes, we would say, thank you for volunteering, but we can't enroll you. If they said no, on that day of vaccination, they got a blood draw and they got a nasal swab. And that is how they determined who had either current or previous infection with COVID, and they didn't know it. So the breakdown was 8 cases in vaccine, 162 in placebo, and that gives you a vaccine efficacy calculation of 95%, with a 95% confidence interval that's pretty tight, 90 to 97%. This second group here looks not only at people that did not have evidence of infection in the past, but it also includes those people who did have evidence of infection in the past. And when you look at that now, because this gets to the question of that some people had of, well, wait, what if somebody's already had COVID and then you vaccinate them? Could they have a severe reaction to vaccination? Could they end up having, if they got reinfected, could they have more severe disease? Well, what the data looks like is we only had 9 cases in this group, 169 cases in this group. The efficacy was maintained, as was the safety profile. So those are pretty good numbers. If you kind of break down efficacy according to these groups, now the study was not powered to look at all these different subgroup analyses, but we wanted to show the data because we think it's pretty compelling information. So regardless of age group, except for this greater than 75-year-old age group, because there just weren't enough cases, regardless of age group, vaccine efficacy is maintained greater than 90%. When you look at race or ethnicity, again, it's maintained. But again, I take a little bit of caution and I'm a little bit measured when I see big, huge confidence intervals like this and small numbers over here. So African Americans, 0 cases in the vaccine group, but only 7 cases in the placebo group. So you get 100% efficacy, but again, you got to take that with a grain of salt. And then, there was a little bit of variance in terms of efficacy across countries, but again, Brazil, which had the lowest, it only had 8 cases or 9 cases. So this is my favorite. This cumulative incidence curve. So what you're looking at is the cumulative incidence of infection. Over here is the days after dose 1. So remember, everyone gets vaccinated their first dose at day 1, their second dose at day 21. The blue line represents people who got placebo, and every single one of these X's, squares, is a case. If there were more than 1 case on that same day, they would just give you a single square, it's just overlapping. And then on the red is, these are the people who got vaccine. So what we see here is very early on, we see about day 10, we see these curves start to diverge, and they keep diverging because the people in the placebo group continue to get infected and get COVID, and the people in the vaccine group are protected. If you look down here, what you see is everyone's asking, is there any kind of efficacy after a single dose? when you look at efficacy between dose 1 and before dose 2, you had 39 cases in the vaccine group, 82 in the placebo, efficacy of 52%, which if you're using influenza as a benchmark, the 2019-2020 influenza vaccine efficacy was calculated at about 45%. So this is a very comforting curve. Here, they use the Charleston Comorbidity Index to kind of break out all the different comorbidities that people who volunteered for the study had. So we enrolled people that had stable HIV, hepatitis B, and hepatitis C. So that's why you have people with HIV, malignancies, et cetera. And this just is to represent that we did enroll people that had medical comorbidities, because those are the people that are at greatest risk of a bad outcome. And so you need to generate safety and efficacy data in those groups. And then when we look at the adverse events, again, the vast majority of adverse events, so any adverse event, 26% in the vaccine group, 12% in the placebo group. The investigators had to assign whether or not they thought it was related. You see about a fifth, 1 out of 5 thought that these any events were related to vaccine, and about 5% in the placebo group. But again, in terms of severe or life-threatening, very, very few numbers. There were a total of 6 deaths, 2 in the vaccine group, 4 in the placebo group. And I actually think I've already. Oh, this just looks at a breakdown, a further breakdown. This is from the supplement in the journal article, looking at efficacy according to comorbidity. And again, the whole take-home point is there is maintained efficacy across different age ranges and across different comorbidities within the cohort. And then, this was a big thing as well. So there is, because of the inactivated RSV vaccine from a while ago, that kids who participated in that trial, they got the vaccine and then they got a natural infection, and there were a number of them that had worse disease after vaccination than those kids who didn't get vaccinated. And so there was a concern, and we had some animal data with SARS-CoV-1 and MERS-CoV, that there was potential for a similar, depending upon the vaccine construct, that there was some potential. So the regulators and scientists have been pretty concerned about where are the severe cases occurring, and is there any evidence that the vaccine is actually making natural infections worse? The numbers are good. It's one compared to 9, but really those are very, very small numbers. Moderna, which has a very similar vaccine, a messenger RNA vaccine, they actually have all of their severe cases in the placebo group. So I think there's some trends, but it's they have to keep collecting data. All right, so there's a lot of remaining questions. So they vaccinated, they enrolled 44,000 people. They followed them at a minimum for 2 months, but the idea is, what if if you start rolling out millions and millions of doses, are there rare but significant safety issues that we didn't see in this initial trial, but may happen further down the road? But that's why the trial is going to be 2 years. Of course, the idea of long-term safety following a natural infection, we have to look at that. Safety and effectiveness after a single dose, and 98% of people got their second dose, even more. So it was a little difficult to study this, but this is something that people have questions about, especially trying to roll out in a pandemic a 2-dose vaccine. Does the vaccine prevent asymptomatic? Sorry, that should not be disease. It should be asymptomatic infection and limit transmission. And then, of course, safety and effectiveness in the groups that either were not represented in the trial. So this would be pregnant women, or women who are breastfeeding, or people with immunosuppressed conditions. But also those subgroups that were included, but the numbers are just not that significant. So in summary, I think the Pfizer trial indicates that there is the potential to protect people from COVID through vaccination. I think that a vaccine combined with improved public health interventions, masks and social distancing as the foundation, that could bend the epidemic curve significantly. But, you know, a rollout and driving vaccine uptake are both going to be heavy lifting and are both going to be challenging. And, you know, we have more work to do. And then based on the currently available data, you know, it's really expected that more than one COVID vaccine will be made available in the US, maybe even before the end of 2020, because Moderna's date with the FDA is next week on the 17th. But certainly by the end of 2021, it's not inconceivable that we could have 3 COVID vaccines being made widely available under emergency use or through a full license. Okay, so thank you very much for your time. I will stop sharing.
Well, thank you, Dr. Thomas. I have to say that was a very comprehensive overview, and there haven't been many things that have made me feel as optimistic in 2020 as that cumulative incidence curve. I know we were looking back at, you know, an interview that you did this summer, and I think even you were not totally optimistic there'd be a vaccine developed by the end of the year. So today must feel pretty great.
Yeah, cautiously optimistic.
I have a couple questions in the chat I'm going to ask. One of the first ones that was sent to me, because we do have a lot of healthcare workers, obviously, that assume they will be in the first cohort of individuals getting a vaccine. Should a medical professional that receives the vaccine, should they be worried about shedding to vulnerable, high-risk family members?
Yeah, so this is not a replicating vaccine, and you're not all you're making is the spike protein. That's all you're making. So yeah.
And is history of ACE angioedema a contraindication?
So if an EUA is issued, the FDA will meet with Pfizer to then agree upon what the label is going to look like. And in the label is where they have all the contraindications. I can tell you, you know, in the Pfizer study, we did not have any of these acute hypersensitivity, either anaphylaxis or anaphylactoid type reactions. But they really have to, you know, those two people in the UK that had severe allergic reactions is going to require them to go back to their database. I mean, I think in the end, what's going to happen is we're going to have the data that we have, and we're all going to do the job we've always done, which is have a conversation with our patients who may be eligible for the vaccine and to determine risk benefit based on their history of allergies, and what their risk of infection and or a bad outcome from infection could look like. It's always, you know, every vaccine is going to have the label that if you have a known allergy to any of the components in the vaccine, you should not get vaccinated. But
and a recommendation for patients who are on immunosuppressants or steroids?
Yeah, so we would not allow people who were immunosuppressed to be part of the study. I mean, we did enroll people that had hepatitis B and C and HIV, but they had to be well controlled. They had to be well controlled. So yeah, I don't think they're going to be in the first round of vaccine recipients.
All right. Well, I know that we could all dive into this quite extensively and keep you here all night, but I am sensitive to everyone's time. So we'll have to have you back for a full session. And if folks do have questions, keep putting them in the chat, and I will share them with Dr. Thomas after this presentation, so that perhaps, you know, in between appearances, you might be able to shoot us back some answers.
I'd be happy to.
Thank you so much.
Thank you.
Yes, I agree. I appreciate the presentation, Dr. Thomas, and I look forward to hearing you again in the future.